As I said, we'll discuss towards the end, the next 30 minutes or so. So we learn about, from Frank's talk about the clinical, from a medical clinician standpoint to educational ecosystem with multiple countries training through this collaboration and also industry perspective and outlook of the critical quality attributes. And then you can see the differences between a larger nanoparticle versus a dendrimer. You cannot use the same equipment. Some equipment don't work at 10 nanometers in size, and some equipments do. And those that have, beyond the size and measurements, it is also the drug release kinetics and stability and so on, to regulatory science and workforce development, and then finally valley of death. How academic research, really most of it doesn't get to translate into clinic, and then there is NCL and EU NCL that help, at least in US for cancer, and EU NCL for diseases beyond cancer. Some help, but there are many challenges. So this is excellent. We have different viewpoints, and so I'll open this for questions first before. Any questions? Yes.
Is the key reason? So is it the key? Is the key reason that there aren't that many nanomedicines in the market that the ROI just isn't good enough? That the upfront costs are higher for nanomedicine than a drug? I mean, it's what Marianne was alluding to, I think. I mean, there aren't really. There's $1 billion euros spent on research that was said this morning, but you could spend that on one project, couldn't you, to get it to the clinic?
Let Marianne respond. Have a mic to Marianne. Oh, maybe.
So I'm not sure I'm the expert on this, but I just go from I guess, perspective and discussions internally. One, attrition rate is high in the industry anyway. I think it's probability of success is running about across the industry 8%. I think that was reviewed, Nature Drug Discovery Reviews earlier this year. So a lot of what we do doesn't get anywhere anyway. So if you're backing something of high risk on a nano and it's costing you, the comment from the head of oncology to me is I can have 5 programs for one of yours, Marianne, one of the nanos. So I think there is a little bit of a work for us to get things a lot more cheap and slicker. And I think the first time you do something, it's harder. And we are only in phase 2. We haven't presented anything to the FDA or EMA yet. So I think, just to answer your question, I think they are more expensive, so you have to then choose your target. A lot of people in nanos are doing what I'd say Me Too targets, or waiting for generics to come off, and then you've got to, which is difficult because there aren't the drugs available, and then you've really got to beat what's out there. And if you're competing with docetaxel, what's it, $2 billion, you've got to be really good. That if you do it earlier, you've then got a risk that your target isn't clinically validated, so therefore you're adding more risk in terms of risk and something more costly. I don't know, have I answered your question, or just not exactly, but...
The number was put up, we spent €1 billion in R&D this morning on the EU lady, which seems like an awful lot of money. But just for one of your programs, that's what you're probably spending that kind of money to try and get those 2 targets to market, aren't you?
No, that's not $1 billion.
To get them to actual market?
Maybe all the way through clinical trials.
Yeah.
So if you're adding phase 1, phase 2, phase 3 clinical trials, multi-center trials,
I don't think it's quite...
it is quite that high.
Yeah, it's 100 million from phase 3, isn't it?
Phase 3.
Yeah, I haven't
It depends.
It's cheap then.
Maybe Frank and...
Yeah. Well, I guess there are different analyses, if I may dive in, but the dark picture I painted with the cost to bring something to the market is essentially an average that was published, I guess, now a year and a half ago, and I forgot exactly the resource. But the debate is out there, and whether it's $500 million or it's $2 billion is not so important. I think it's too much money for small inventors and for medium-sized companies. And with that, and as we know from large pharma, and we shared actually at some point the same company that we worked for, it's inhibitory to some of the innovation processes, and that's something we need to do collaboratively. And that's where we need to think how to do this better, differently, and in a consortial way. And I think the opportunity to leverage those areas where the costs are the highest, such as clinical development throughout the phases into phase 4, I mean, that's where a lot of the money gets really spent. And I don't know, the data there, of course, ranges from, I don't know, 300 million to about $500 million or something like that, just in the latest phase of the clinical trial. So that's something we need to address. And the opportunity is there now, because also it is time to innovate. We are in the midst of the digital revolution, and innovation and making better products and inventions, the time has never been as good as now, through the connectedness. And you will say, well, that's a little bit over-optimistic, and yes, I cannot predict when exactly this will graft and take, and when we really will see those changes. But I think the opportunity to link data, understand data, connect data, liberate health data, leverage them in the early phases of clinical development in a societal way, in a consortial way, that will save significant resources. Systems biology, data linking between research databases and clinical databases, and characterization and preclinical development, animal models and microphysiological systems. Also exploring further the dog genome and the targetability of the dog genome for diseases, defining diseases in a different way and coming towards personalized medicine. This is all opportunity that we have in front of us, and I don't think we should shy away to innovate, but it requires a consortial approach. I don't think a single entity can do that. And if you just think of ownership and, let's say, profit, I think you can't make that leap. You have to rethink what we do in our society as health professionals and researchers. And there are simple things that we can do first. Data access, liberation of health data, simple things like unique global personal identifiers are missing for patients. We are still pouring data from different data sources, and we get dirty data just because we don't know where they come from. And also tomorrow I'm going to talk about simple things like the source information for clinical laboratory data. That's, first of all, not interoperable, can be coded in many different ways, and creates a lot of confusion when you actually look back into the evidence. So the traceability to source data isn't there. And if we don't solve that, we will not make much progress in time that we need to make, and not at lower costs. And again, the research judgment is out there to what and how high that impact can be, but I believe those are really important components. Thinking back and talking about consortial product development, I think with some exceptions, that's probably one of the major modes of operation of innovation.
Thank you, Frank. So there is one question. Yes.
Yeah, I've got two scenarios. I just want your comments, perhaps from Marianne. There are drugs which go all the way to pharmacovigilance, and then at that point you realize perhaps there's cardiotoxicity or whatever the case, and you improve bioavailability or the toxicity by using nano formulation. When you do that, how do we commercialize? Do we go back to phase 1, phase 2, phase 3, or do we just do phase 3? Or how do we react there? That's one. The second one, in my group, we were able to encapsulate TB drugs by lipid nanoparticles, and we could not see the concentration so much on the plasma, but the efficacy was there. So it was difficult to confirm, because you can see the mice were being killed more effective than the encapsulated nanoparticles, encapsulated drug, but we could not see that amount of drug we're expecting to see in the plasma. We didn't know what happened, where was the drug, where was it efficacious, but still it was not in the blood. So I just want some comments on that.
This is for Marianne.
I'll take a step. I'm not sure. So I guess the first thing was you mentioned on PK. So I've had a lot of heated debate, I'd say, with colleagues on PK. PK departments are set up to measure plasma profiles, and that's meant to be a surrogate for efficacy and potentially safety. And when you start targeting and do nano delivery, it doesn't really mean anything. And total drug, if you've got tissue accumulation, your plasma PK doesn't really mean anything. So I think I would err a lot of caution when looking at systemic, plasma PK measurements, and total and free drug are really important. And a lot of data, people, I've been handed data, here's your free drug, and then we've done a bit of maths on it, and you've got 300% bioavailability. And I kind of say, nanos are clever, but we're not fabricating material in the body. The bioanalysis must be wrong, or I've got a validated technique. So there, I guess, two things there. One, I don't think PK is a great surrogate anymore, so maybe you need to look at PD or efficacy endpoints. And two, be really, really careful about total drugs and free drugs, and make sure the numbers add up. So that's the second question. Do you want to come in on that while we do that question?
Yeah, in support. It's a question also of compartmentalization, and those more complex drugs, as we call them, and specifically encapsulated, they don't just follow a simple PK profile, and I think that's one of the challenges that has to be considered.
Yeah, so I think
Practical
the
advice, of course,
question
can be in many aspects, and I think that goes beyond the forum here, but I think we have a lot of experts here who can help with that to get your specific questions and the particular compartments sorted out with different technologies.
You want to comment on that, any of you? Yeah. So, I can tell you one thing. For example, I'll give you a classic example. Hezi is not here in the room, so I can say doxil. Liposomal doxorubicin. So you have, the doxorubicin hydrochloride is approved, but then you encapsulate into a liposome. You get away from cardiotoxicity, that's the bottom line, but it's a new chemical entity, right? So then you still have to go through the whole process, phase 1, phase 2, phase 3, because the reason is that if you have, the half-life of doxorubicin hydrochloride is completely different from half-life. Biodistribution may be different. So you may have unintended toxicities that you don't know of. And so any new chemical entity, it has to go through. Similar to the other famous example is paclitaxel and Abraxane. So Abraxane is a formulation of albumin with paclitaxel. The active ingredient is the same from Taxol, which is a chemophore formulation, but they still have to go through the clinical trials because now you change the formulation chemical entity. So one more question at the back.
Yeah. So I have one question. Some of these nanocarrier release the drug more slowly. They form the tissue depot, and when we do the PK, obviously PK is nowhere because it has gone down. Now, in case you have to repeat the dose as we do with the main formulation, so how difficult it is to really determine the dosing, because it's PK-wise, it goes down, half-life is very low. Tissue-wise, it's accumulating quite a bit. That kind of situation, how do you go about it?
Any speakers? Any comments? PK? I'll come back to the education and workforce questions. Okay, go ahead.
So I think one of the considerations is to have a good clinical endpoint or surrogate endpoint. You need a biomarker. Otherwise, it's very hard, and you cannot define it just in conventional. We just touched upon this. And of course, again, there are different options, it's good to have a correlate and to validate it. That's probably the best way to go.
So let me ask these questions. I have a lot of questions. I got really excited, took a lot of notes. So one question I have maybe I'll start with Marianne, because that's the closest in clinical development. So the question is, and I lived in this nano field for some time, few decades, and it's always, you do all the preclinical development, you're a university professor, you're excited, you start a small company, the big companies don't come because they don't have experience, expertise, the comfort level to take something that is new. Right? If it's another small molecule or biologic, depending on the company profile, they immediately, they understand how to, what the pathway. So for you at your company, so you said something about, okay, the traditional chemist, small molecule, they know what to do, but then now if you take a dendrimer with a covalently coupled drug, API, the analytical procedures are completely different. The purification procedures, the synthesis procedures are all different. So how did you train your scientists? How did you, it's not a comfort level. I was a synthetic chemist, small molecule synthetic chemist before I became a dendrimer chemist. Very uncomfortable situation. And then from there, getting into nano is even more uncomfortable because it's more polydispersed. So how did you train your scientists, workforce?
Yeah, I think it's a difficult question. I think initially we tried to sell the concept of nano or in terms of helping therapeutic index. So we picked on initially one drug that had gone into the clinic, so that was a aurora kinase, that was going to go ahead in phase 2 in AML, but had come out of solids in phase 1 because of therapeutic index, and kind of sold the well, if you wrap this up in a nano, and that's what that will help. And I guess in the end, that didn't happen because the nano is doing both. But I guess we sold it as an idea and a concept and a project concept to the business. And it took time and effort and a lot of championing, I would say. But once it comes in as a project, we have to develop it and we have to, it's there. So we then bring people in. So we've kind of ups. So it's similar for the dendrimer. And synthetic chemists and process chemists, don't really understand polymers, which shocked me because I'm not a chemist, and I was telling them what a branch polymer and but I think they kind of liked having something. Some people like having something different and challenging to do. Other people find it really stressful. So it's about managing and working and supporting those that want to do it, and also supporting people that are really outside their comfort zone because they're used to making tablets and now they're in a group called Advanced Drug Delivery and they're expected. We've got mRNA coming along with LNPs. That's even scarier for some people, and it's lovely. So it's around that personnel piece. I think we came a bit unstuck. We were working with Bind and they were doing everything, and then Bind, unfortunately had some political issues, so we had to adopt internally quickly to be able to supply and understand and support the process development. So so that involved working with management to get extra capital for bits of kit, etc. So I'm not sure, hopefully I'm answering your question. It happened with the champion, the projects, reason to believe that this was a good way to go, and then we're bringing people in, and now we're also bringing and actually going out and developing people, reskilling people and bringing people in with different skill sets in terms of polymer chemistry, in terms of intracellular drug delivery, imaging, etc.
Okay, so, yeah, that makes sense. But it's not an easy thing. The transition is not easy. That's why I was kind of Frank, you want to say something?
Yeah, actually, also to your point of what kind of education and preparation, I think the session is called, I guess, more focusing on education and
Workforce.
But workforce, but it's really about knowledge management. If you think about it, knowledge management is changing in a significant way, where standard of care is merging with clinical experimentation and clinical development, but also in a way academic training is merging with research, clinical development, product development, market research, post-market information, epidemiology. And to manage that whole enterprise of knowledge in particular areas, I think we have to rethink how we do it. And it's unfortunate that we haven't attracted some of the other folks today, because maybe even because of the title of the session, I'm not sure. But it's all linked together, because those issues that we find in review, the review questions that end up being scientific questions when a new submission comes to us, how do we deal with it at the agency in a way, in a consortium way? Because we start research in a particular area that often includes the stakeholders, the developers in some ways, but also academic experts and scientists all over the world to address this particular question. And that, of course, leads to new educational material. Not only, it leads also to publication of scientific literature, and ideally it will lead to the information that is required for the review process. And let's be honest, we all, and particularly in our organization, have issues with that. But we need to work together to come to better platforms, to better information platforms, to real-time access to information and research outcomes. And in a way, again, I say it, the consortium approach to innovation. And that, I think, is something that we can do.
Okay. So let me ask you, Jack, about the international, he's talking about international consortium. And so this is a classic example. I see India, Israel, a lot of countries, and you're training the students. So who is teaching these courses? Meaning, are we training the workforce in a proper way?
Well, the idea is to share knowledge and experience. We are, in the consortium, we have teachers teaching Nano, researchers, and they are already teaching courses. And they are sharing their courses that they are teaching, and we are aggregating a pool of courses. And in this way, we can learn from each other, from different cultures, and it's become a sort of sandbox where you put your knowledge, your expertise, and you share it with other partners. So it is a win-win situation, as I see it. We not only gain and share knowledge on nanotechnologies, but also on learning technologies, infrastructure, and everything that is connected with the dissemination of knowledge, and also pedagogical approaches like putting the student in the center, and maybe some discussions about the role of the teacher nowadays, that they don't have the monopoly to knowledge, and the knowledge or the information is available ad hoc any place, and then you don't need to teach students information or memorize things because it's not relevant anymore, but have to critically retrieve information, have to be innovative, and things like this. So when we put all this in a platform that is transparent for all the partners, then we have the impact of sharing knowledge and learning from each other.
Okay, that's great. And I have a question for you. So my question to you is, you talk about the bridge, the gap.
Not a bridge yet.
It's not bridges. So my experience has been, yeah, you have a question?
I wanted to say something,
Please.
but maybe a little bit later.
Okay, so I will quickly ask this question, then we'll come back to you, because this is very critical. Most of the academic work, I don't want to slam on the academics, and I was an academic before, but then when I was at NCI trying to translate things into clinic, beyond a proof of principle, we cured cancer in mice, we want to go into humans. Most of the science is irreproducible. in the nano area. And NCL has lessons learned workshops as to, what are the failures, and dendrimer, one of the best dendrimers, didn't make it because it is safe but not efficacious. So, but then when I talk to the faculty, they always say, well, I just want to do research, publish papers. I'm not interested in getting into clinic, right? And they don't have to. But my bottom line argument is, well, you have to do reproducible science, right? It doesn't matter whether you get into clinic or not. And that's where this challenge is for nano, because if it's a small molecule, that's not a significant problem. So what do you think about the academics, getting, at least in the nanotechnology area, is there a way that can happen?
There's a movement now for, the lack of reproducibility isn't just in the nano area. It's all over the place. I mean, Amgen did a study and found that much of the data that they got from academia was not reproducible for one reason or another. And I don't think there are very few people who would say that there's a lot of fraud involved there. I think there's a lot of sloppy lab management perhaps involved, where people just, it's being done by graduate students on equipment that is not always top drawer. There's a lot of lab discipline that is not part of our culture. And I think going to that culture is an anathema to some scientists who feel it will inhibit their creativity. So, on the other hand, as somebody I know very well says, no patient was ever made well by a journal article. And that may be an exaggeration, but I think what he's trying to say is that at some point when you take public money, there may be a responsibility to think beyond looking at the lab as a sandbox where we get to play. And we have to sort of think about how we're going to manage this process, not just at the lab level, but at the university level. The universities provide, at least in my experience from a few, provide very few resources for helping to establish the kind of facilities and infrastructure that are needed to really be confident that you are creating quality data. And the divide is getting much, much bigger. So for example, toxicology data to go into the FDA from a company point of view now has to be done in SEND format, which is, a specific nomenclature for your electronic records so that the data is interoperable. But if I were to go to almost any university in the US and ask them if they know about SEND format, even when they're doing toxicology studies, they'd probably look at me blankly. And it takes 2 days training program for most people even to learn SEND format. So who's going to pay for that? Nobody pays for any of this education. You get as far as discovery. After that, there's no education money at all in the system.
Scott?
Just to add to that, I think I agree more broadly that rigor reproducibility has been a major issue, and NIH has certainly prioritized that. And I think a number of institutions are starting to implement programs for the trainees. It's just for the existing researchers, I think it's a bit of a challenge how that's going to be, that awareness, and we don't usually say educating current faculty, but how that's going to be disseminated. And on the quality side, we do, for the clinical and translational science institutions in the US, they do have generally a requirement to have a regulatory knowledge component, and there's usually someone responsible for quality there, but it's a pretty limited resources to try to address a much broader issue.
Yeah.
Well, I'm a bit hesitant. I have heard now this session, and I think there is such a lot of helpful possibilities that are there for further education, for development, for solving. Maybe you are right at the university in Basel or at our lab. I ask always the people, did you fill in your logbook? So I agree with you that this is a big problem. Now you talked about Zen format. I don't know what it is even. But what I wanted to say here, I see so much quality of people that are knowledgeable in the field. But I ask myself, and I would like to speak also that, for instance, ETPN has a hub for translation. I haven't seen any product except for what Nanobiotics did coming out of that hub. And my question is maybe totally wrong, but I ask myself, if nanomedicine is doing its function, and if we are going to CLINAM and we see so many attempts to solve problems, like we have heard them this morning, why is the pharmaceutical industry then so hesitant to make a pool and to invest? If you would have 50 companies that are doing pharmaceutical drugs daily, who know the procedures, who know how hard it is to go to the to get approved, who go through phase 1, 2, and 3 in routine. Why would these companies, or why can we not, with all our knowledge of education and further education and development of students, of keen young people? I mean, yesterday at 5 o'clock we had this room full and people were standing for the talk of Avi Schroeder. I was really fascinated that on a Sunday at 5 o'clock you get 78 people from students from anywhere in the world into a room to learn about this. What would be the possibility to motivate those who are already making tablets, maybe to come a bit away from Avastin for 63 sorts of breast cancer and 18% react on it and the rest not, and just selling it, or to invest money into, I mean, an initiative, a human initiative would be, could we try, can we, with our knowledge, motivate companies to set into this technology, and not calling it maybe nanomedicine, but just say that we are novel methods, novel attempts, and immunology using in a much more aggressive way to come to products. Because I fear that if you look at nanomedicine, after 11 years doing CLINAM, I am allowed to say that I did not see so many products coming up. However, there is a company downstairs that is exhibiting, it's Inomedica, and they have brought their product as far in 5 years that they are almost going to get an approval. So, I mean, I hear always people talking about that it costs 2 billion and that we cannot do it and that it's so difficult. But we have examples of people who had luck, who did it, like Chezi with the crystals in the lipid, like Desai, who unfortunately couldn't come. So I have a problem. I have a problem to listen to this, which I think is excellence, and the proposals, or if I look at the slides of you, Jack, I think this is fantastic. But I always, everywhere in the sheet see will. It's always future. We are going. We will do. I would like to sometimes see a system working.
Yeah, but it was in the previous project. This is ongoing projects that I displayed.
So, yeah, so that's a good point. So maybe what I suggest is maybe in the next meeting, you bring all the pharmaceutical companies in Basel together. And
They don't come.
so,
The problem
yeah,
is
so and
they
you have,
don't come. Novartis is an exception.
so FDA is coming, EMA is coming, all the regulators are here,
Yes,
right? Okay, so
but when I talked to a person in Roche belonging to the uppermost board, and he said, "Listen, if we need FDA, we fly them in or we go there. We don't care.
right, but my point is,
We don't come.
so
don't
we
care."
have regulators here, right? We have IPRF meetings, and we have most of the regulators at this meeting.
I know.
We come to this meeting because we like the meeting. The quality is excellent, right? Now, the challenge is, and that's why I was asking the question, it's a comfort level for the companies to invest into something new, right? And that's I think from minimum experience, I've only dealt with maybe 50 companies, small companies trying to, get investments from larger pharmaceutical companies. There is the comfort level. If it's a small molecule drug, again, biologic or something else, they know how to do it. So if it is something new, then only a few companies are taking the risk. But the good thing is we have data from FDA, and we present this. Catherine Tyner probably presented in the other session. I'm going to show one graph, and you have seen that graph, is that there is a gradual increase, very, very gradual increase. And then what makes me very positive is I see two dendrimers. One is a presentation here. The other is, I think Don is talking about in the next session about phase 3 clinical trials of a dendrimer, which when I worked in the dendrimer field, it's a nightmare in terms of how to get these products into clinical trials. And then I'm happy that I see these not just polymeric and liposomal formulations, but then there are newer ones that are coming in. So you have a comment.
I'd like to say is that many of these companies are very closed-mouthed. I mean, you have to understand the big companies have to be very circumspect about the products that they're working on. They're not going to talk about them necessarily. And they may be investing at a low level in small companies to de-risk the products in the small companies before they take them in. So, when you talk to some of the big companies, I think there's more going on than we might think. But they may not be wanting to talk too much about it because their stockholders are very risk averse. So you don't want to sort of ring that bell and then find you have to unring it.
So I also think the tide's changing a bit. I think a lot of companies were into high throughput screening and working with kinases and small molecules. I think many companies have got a lot more diverse portfolios now with antisense, oligos, mRNA, small molecules that have been intractable because of therapeutic index. So it feels, and certainly from internally it feels, going out to meetings, because of the diversification, we will need more advanced drug delivery and more nano. And certainly for all the nucleic acids and peptides, we'll need nanotechnologies to get into cells. So, looking at the portfolio in 2012 and now, completely different. And it's not, that's us. I know, other companies are in the same boat. So I think there is more activity. I agree there are less big pharma supporting some of the more specialised nano meetings. But I'm very hopeful, Beat, that it will change.
So, Beat, don't be so impatient. You already contributed to the change in the tide, and I agree. And the other part, in terms of the access to the knowledge and the information, it'll change. And just to give you the other part of this equation, the hesitation, but large pharma also realizes to some extent the structures are sometimes too rigid for innovation. That's why they moved into the Boston area. Why? Because there is enough bubbles and innovation that stimulates actually large pharma. And then, of course, it's a question of confidence. You have to calculate the risks and the investors, and so on. So it's coming. It's moving forward, and with the increased access of knowledge, it will move faster. There is no question. Of course, it's difficult to say when it's going to really break through and when we will have more than half all of the product being nanomedicine and so on. It's hard to predict. but it's on the march.
By the way, when you speak about trust, in 2010, I was in England and I said we need a European one-pipe characterization lab, analog to the possibilities that we see at Scott McNeil. And the answer was, we will do it, and it was picked away before we even knew there was an initiative called European UNCL. And the problem is that you can talk to anyone in industry, he says, we will never, never, never go there, because I don't want my findings to be either first in Münster, then in Rome, and then at EMPA in Switzerland, and then at 2 other locations, and then getting it back. Therefore, we have now in Switzerland launched an initiative to propose to the state of Switzerland to make a one-pipe lab for whole of Europe in one place. So I think when you want to win industry to use such things that we are mounting, I think it must be more bona fide than a network where your product goes on a trip. What do you think about that?
Well, I think Anil has
No,
personal
Marianne.
practical. So, sorry.
Yeah.
Marianne,
Marianne.
would you, Marianne, do that?
I seem to be put on the hot seat a few times here. We haven't got a problem with the EUNCL. We're collaborating with the EUNCL, and it's not a problem.
It works.
I think it's sort of too early days, ongoing. But I don't think, the terms of the agreement are really important about who owns the IP. We're not going to want to give IP of our drugs to other people, whoever we work with. But as long as it's all confidential and the agreements are in place, I don't think that's come out as an issue from the discussions that we've had. I'm probably being too open.
So we hope, Bia, that you will bring the pharmaceutical companies to the table next year, and we'll continue to have this discussion. So we ran out of time, but thank you again for all the speakers, excellent talks, and then we learned quite a bit, and then excellent discussion. We'll close that session. Thank you. 2020. Okay. Well, thank you again.